This video will explore the structure and function of the cell, the fundamental unit of life. Cells are the fundamental unit of life and are self-replicating structures. They were first observed by Robert Hooke in 1665. Organisms can be unicellular or multicellular, with cell diameters ranging from 2 to 120 micrometers. The cell theory, proposed by Schleiden and Schwann in 1839, states that all living things are made up of cells. Cells are the smallest working unit of all living things, and all cells come from pre-existing cells through division. The different substances that make up a cell are collectively called protoplasm. It is composed of 70 to 80% water, carbohydrates, lipids, proteins, and electrolytes like sodium, potassium, and calcium. The major subcellular structures present in a cell include the cell membrane, cytoplasm and its organelles, and the nucleus. The cell membrane is a thin, pliable, elastic, outermost structure that envelops the cell. It consists of a bilipid layer with embedded integral and peripheral proteins. Functions of the cell membrane include protection, digestion, and selective permeability. Non-polar molecules move rapidly, while polar molecules penetrate much more slowly. This property helps maintain components in the intracellular and extracellular fluid. The cell membrane also links adjacent cells together by junctional complexes to form tissues. Furthermore, it acts as a dielectric material, giving it a very high insulating value. The cytoplasm and its organelles are critical components of the cell. Cytoplasm is a thick, gel-like, semi-transparent fluid found in both plant and animal cells. It consists of the cytosol, cell organelles, and cytoplasmic inclusions bounded by the plasma membrane. The cytosol is the aqueous part of the cytoplasm outside the organelles, accounting for almost 70% of the total cell volume. It is a gelatinous substance made of cytoskeleton filaments, organic molecules, salt, and water. Organelles present in the cytoplasm include mitochondria, endoplasmic reticulum, lysosomes, Golgi apparatus, peroxisomes, and vacuoles. Each is bounded by a lipid membrane and has specific functions. Mitochondria, known as the powerhouse of the cell, were first observed by Kolliker in 1850. They are filamentous or globular structures with lengths between 5 to 12 micrometers and diameters of 0.5 to 1 micrometer. Mitochondrial components include the outer and inner membranes, the intermediate space, cristae, and the matrix. These membranes are composed of phospholipids and proteins. The outer membrane contains porins, allowing small molecules to pass. The interior matrix contains enzymes for the citric acid cycle and respiratory chain oxidation, involved in the oxidation of carbohydrates, lipids, and amino acids. The inner membrane contains ATPase and other enzymes for ATP synthesis, along with enzymes of the electron transport chain. These mechanisms enable oxidative phosphorylation and ATP synthesis. Mitochondria function as power-generating units, maintain calcium ion concentration, facilitate energy transduction through respiration, and are responsible for thermogenesis. The endoplasmic reticulum is a network of tubular and flat vesicular structures in the cytoplasm. It is an extensive network of closed, flattened, membrane-bounded sacs called cisternae, filled with endoplasmic matrix. The endoplasmic reticulum comes in two types, rough ER and smooth ER. Functions of the ER include protein synthesis and segregation, unsaturation of fatty acids, and muscle contraction, where it is known as the sarcoplasmic reticulum. Golgi bodies are a collection of membrane-enclosed sacs stacked near the nucleus. They consist of 4 functionally distinct regions: the cis-Golgi network, the Golgi stack, and the trans-Golgi network. The Golgi is the cell's, wrapping and packaging department. It produces secretion granules, packages proteins, and is the site of lysosome formation. It also adds carbohydrates to form glycoproteins. Lysosomes are irregular structures, 250 to 750 nanometers in diameter, surrounded by a unit membrane. Their interior is acidic, maintained by a proton pump, and they contain cell hydrolases that function best at this acidic pH. Lysosomes act as the cell's digestive system, degrading macromolecules, worn-out components, and exogenous substances like bacteria. Because their enzymes cause autolysis when a cell dies, they are called suicidal bags. Peroxisomes are about 0.5 micrometers in diameter, surrounded by a lipid bilayer membrane. They have a structure similar to lysosomes but with a different chemical composition, sometimes containing a urate oxidase crystalline core. Peroxisomes contain oxidases that produce hydrogen peroxide, Catalases degrade this peroxide to yield water and oxygen. Proteins are directed to peroxisomes by a unique signal sequence with the help of protein chaperones called peroxins. Functions of peroxisomes include H2O2, metabolism and detoxification, photorespiration in plants, and biosynthesis of lipids such as cholesterol, dolichol, bile acids, and plasmalogens. The cytoskeleton is a system of fibers that maintain cell structure and permits changes in shape and movement. It is primarily composed of microtubules, intermediate filaments, and microfilaments along with anchoring proteins. Microtubules are long, hollow structures that determine cell shape and are involved in chromosome movement and ciliary motion. Intermediate filaments provide scaffolding and resist external pressure. Microfilaments, comprising actin, are responsible for cell motion. The cytoskeleton is involved in the movement of chromosomes, cell movement, the movement of secretion granules, and the movement of proteins within the cell membrane. Next, we will discuss the nucleus, a critical component of the cell. The nucleus contains chromatin, RNAs, and nuclear proteins. It has an internal structure that organizes genetic material and localizes nuclear functions. A matrix of nuclear lamins extends into the interior, organizing functional nuclear bodies. Nuclear lamins serve as sites for chromatin attachment. Chromatin is organized into large DNA loops, with specific regions bound to the lamin matrix by lamin-binding proteins. The nuclear envelope is a complex structure with two concentric membranes, inner and outer, an underlying nuclear lamina, and nuclear pore complexes. The outer membrane is continuous with the endoplasmic reticulum. The nuclear membrane is permeable only to small nonpolar molecules. The nuclear lamina is a fibrous meshwork underlying the inner membrane, providing structural support to the nucleus. The nucleus serves as the repository of genetic information and the cell's control center. Its presence allows gene expression to be regulated by post-transcriptional mechanisms, like alternative splicing, and at the level of transcription. To summarize, different cellular compartments perform major functions. The plasma membrane transports ions, while the cytosol metabolizes carbohydrates and lipids. Mitochondria produce energy, and the endoplasmic reticulum synthesizes proteins and lipids. The Golgi apparatus modifies and sorts proteins, lysosomes aid cellular digestion, and peroxisomes utilize H2O2. The cytoskeleton governs cell morphology and motility, and the nucleus is responsible for DNA synthesis and repair. Thank you for watching this video on cell structure and function.